Is it true that as we age, our chromosomes actually get shorter? And is this maybe one of the reasons that we age? In this video, we're going to talk about what happens when DNA makes more DNA or DNA replicates itself. Let's get started. So, in the last video, I talked to you about the central dogma, so I'd like to revisit that just to remind you where we are in the flow of this information. So the central dogma is the flow of genetic information within the cell, and of course we start by talking about DNA. When DNA is used to make more DNA, that process is called replication. When DNA is used as a code for messenger RNA, that process is called transcription. When that messenger RNA is used by the ribosome to make proteins, that process is called translation. In this video, we're going to spend the entire time talking about this step here, replication. That is when DNA makes more DNA. So there are a few models that were proposed of how this might work. So before we really understood this process, there were a few postulated methods. Some people thought, okay, let's start down here, the dispersive model, meaning if this was our original or parent strand here, some people thought, okay, a little bit of this comes up into one, then there's a brand new copy made, and then a little bit of this part goes, and right? This is a dispersive method. A conservative method would say one strand is just basically copied, like you have the original, but then you had a copy, sort of like going to a copy machine, and you get a brand new copy with all new nucleotides. Then there was the semi-conservative method, meaning that because there's two strands in this parent, the idea is that one strand would go in one piece of the new DNA, and another strand would go in the new piece of DNA. Well, what we found out is that that's the winner, semi-conservative. Replication is a semi-conservative process. That is, one strand serves as a template for the other. So each of these, what we call daughter strands, has one strand from the parent. So these methods, conservative and dispersive, were found not to be biologically significant. Semi-conservative is the way it actually happens. So how does this work? So the replication of DNA really is all about the base pairing. So if you think remember DNA, we have this double helix, two strands. Well, what basically happens is the strands open up, and then each of these strands serves as a template so the new nucleotides on the new strand can join. So for example, if this opens up and now I see a G, remember that C always binds with G, and T always binds with A, right? So the new nucleotide, cytosine, is going to bind where there's an open guanine. Adenine is going to bind where there's an open thymine. So basically, once this opens up, the new nucleotides just come in and bind where there's an open spot. So replication this way ensures that hereditary information is continuous, meaning that basically you get two strands that are identical in a way to the original parent strand. So in this video, I want to go through the several processes that allow this to happen. And it's quite complicated, so we're going to break it down into several steps. You should know that there's a large team of enzymes along the journey that's going to help this replication take place. And it actually, what we'll find is that replication takes place in little bubbles. So it's not like we start at one end of the DNA and just copy all the way down to the end. It actually takes place at several little bubbles along the way. So if this is a piece of DNA that we have here, the origin would sort of start here in the center, and it would kind of bubble out from this center part that we see there. So the first thing that happens is that an enzyme called primase comes and primes this DNA, right? So what we know is that we actually can't start adding new DNA nucleotides right at the beginning. We actually need a little primer to get it going. And I'm going to talk about each of these steps in more detail. This slide that I'm looking at now is sort of a general overview of the whole process. We're going to go deeper into each of these. So primase adds a little primer to sort of get us started. Then what happens is this is when we're going to add the new nucleotides. So what you're seeing in this diagram is right, the blue strands that you see are our original DNA. These new sort of orangish looking things, that's going to be the new strand that's complementary to the one that it's next to, right? So in the end, we're going to have 1, 2, 3, 4 strands total because that's 2 pieces of DNA. So what happens in step 3 is that DNA polymerase 3 is an enzyme that builds the new DNA polymer. So it's basically taking the nucleotides and adding them here to the strand, okay? And we're going to go over these ideas, leading and lagging strands, more details in a moment. But basically, one of them, because it can add continuously, kind of leads. The other lags behind because it has to sort of do replication in what we call fragments. Okay? So step 1, we have an origin site. Step 2, we prime it with some RNA primer. Step 3, DNA polymerase starts adding new nucleotides. Step 4, what we have to do is do a little spot checking. DNA polymerase 1 is actually something that's going to come in, it's going to clean up, repair any things, remove the RNA primer, get rid of those, so that we get the correct DNA nucleotides attached to the template strand. Then, because we get some fragments here in the DNA, we need to glue those little fragments together so that our DNA is actually a nice, seamless strand. There's an enzyme called ligase that actually helps that occur. And then, the end, we've got basically 1, 2 complete new strands of DNA that each have one of the original parent strands in them. Okay? So what I'm going to do is I'm going to go through each of these steps in more detail because there is a lot more detail. But if you need to, come back to this part because this is the sort of general overview of what happens. Okay, so let's start with that first step. You may remember that DNA is really coiled up and it's in this sort of tight little super coiled chromatin. Well, the first thing that has to happen is we need to ease some of that coiling a bit. So there's an enzyme called topoisomerase. This enzyme relaxes the coiling of that DNA, so it's not so tightly coiled, so that these other enzymes can kind of come in there and the nucleotides can come in and do the job. You may also remember that DNA is a helix. It's wound around in this helical pattern. So there's an enzyme called helicase that unwinds, or some people say unzips, the DNA, so it goes from 2 and it kind of opens it up, so it's unzipped a bit. So topoisomerase is the enzyme that uncoils, relaxes the coiling. Helicase sort of unzips or unwinds the DNA. And then these things here called single-stranded binding proteins sort of serve as, I like to think of it as a doorstop. You know, if you've got a door and you swing it open and then you like put a little wedge so that the door stays open. To me, that's kind of what these do. They bind to the DNA and kind of stabilize it, keep the door open so that the enzymes can come in and do more of their work. All right, so this first step is really to relax the coiling by topoisomerase and to unwind or unzip the DNA by helicase. So then step 2 is going to be all about the priming. Here's what we know. DNA polymerase 3, right? That's the enzyme that builds the DNA polymers. So that's going to do the most of the work here. That can only extend existing DNA. It can't start by adding new DNA. So actually there's an enzyme called primase that first starts and puts a little RNA primer onto DNA, right? It doesn't make much sense, but that's how it works. So DNA actually first starts with a little bit of RNA primer that then is going to be removed later. And so here's what that looks like, right? So if here's our origin site here in the center, we've created a bubble. And what's happened here is primase, this enzyme that you see there, is adding little bits of RNA primer to this new strand of DNA. Now, once that's done, then DNA polymerase 3 can come in because it's already been started, because it's been primed, then it's ready to go. If you think about primer, what we normally use, you think of paint, right? You're going to paint a wall. Well, maybe a wall is like really red and a really bright color and you want to change it to something neutral. You can't just come in with, like a light gray paint and paint over that red wall. Sometimes you first need to prime it with a first layer so that it's ready for the other paint. In a way, this is kind of the same way. It's a primer. It gets it ready so that DNA polymerase 3 can then add the new nucleotides and build our DNA. Okay, then step 3 is the actual replication part. And this is when we're adding new nucleotides. So DNA polymerase 3, as I've mentioned, is what's going to be building these nucleotides and matching up to the template strands. So if this is one of my strands, right? So think we've got our DNA, we've taken it apart. Here's one side of the DNA, if you want to think of it, one side or one strand of the DNA. So what happens is, as you just saw there, a new nucleotide comes into match. Because this was guanine, and we know that G always pairs with C, the cytosine will come in and bind here. Now, you may remember these are hydrogen bonds that bind the nucleotides together, or the nitrogenous bases together. So then what would be next? Well, the next one would be an adenine. Adenine will come in, bind to thymine. Then, of course, we're going to have a guanine come in and bind to cytosine. And then these just keep coming in and binding until we get the new strand. Now, just a little thing to note. Remember, guanine and cytosine have 3 hydrogen bonds that hold them together. Thymine and adenine have 2 hydrogen bonds that hold them together. But notice what we've got. We've still got our sugar-phosphate backbone, right? Sugar, phosphate, sugar, phosphate. We got the nitrogenous bases in the center. And then, of course, now, because these nucleotides came in, we have another sugar-phosphate backbone over here. Now, hopefully one of the things you might have noticed is that I started with this end because, did you see that this down here is the 3' end of DNA? This is the 5' end up here. DNA is what we call read from 3' to 5'. DNA polymerase 3 reads DNA or adds new nucleotides to the 3 end of a growing strand. So DNA is read 3 to 5. But what you notice here is that this new strand grew from the 5' end to the 3' end. So if I sort of took these back and let you watch it again, we're reading the template 3. Okay, DNA polymerase says, oh, there's a G. I need to match that with a C. But notice this is now the 5' end that's sticking out on this side. So then it goes up from 3 to 5. It reads the T, matches until we get the original strand that we had that is now, as you see, 5 to 3. So DNA is read 3 to 5 direction, but notice the new strand is manufactured in the 5 to 3' direction. Okay. How does this take place? It would seem like all this needs a lot of energy. Well, remember, the enzymes involved are going to help lower that activation energy, but there still is some energy required. In fact, these nucleotides arrive as a fancy word we call nucleosides, meaning they come with their own energy. I like to think of it as BYOE, bring your own energy. So these DNA bases actually come with 3 phosphate groups, which is their own source for energy, and DNA polymerase 3 will bind them. So let's go back to our little single strand here. We're going to add that. Remember, DNA is read from the 3 to 5' direction. Notice that this new strand came in with 3 phosphate groups. But when it comes in and binds, that provides the energy. 2 of the phosphates leave, breaks that bond, provides the energy. And actually what you saw came in was a CTP, right? A cytosine triphosphate. And then what you saw here was, oh, adenoribose ATP, right? That was an ATP, adenosine triphosphate. Once these phosphates leave, that brings the energy for this all to happen, and there's a GTP. You got the two phosphates that are going to leave. That provides sort of the energy to bind these things together. That was a GTP. So what provides the energy? Well, the nucleosides themselves come in with these extra phosphate groups. That provides the energy for this process to take place. Okay, so let's get a little deeper into how this works. Now, I'm going to show you several enzymes here. We're going to kind of review how this happens, and we're going to talk about this idea of a leading and lagging strand. So let's review where we are. This was our origin site. Okay, so topoisomerase, remember, relieved the coiling. Helicase unzipped a little bit. Now, what you got to think is we're moving in two directions. If the origin site was here, we're going to start adding nucleotides in that direction, and we're going to start adding nucleotides in that direction. So in a way, if this is the origin site, we're going to start going in both directions. And so you can see that sort of starting here. Now, the single-stranded binding proteins are going to sort of form that wedge to keep this open for us. RNA primer has already added, RNA primase has already added the RNA primer. And now what you can see here is DNA polymerase III is adding the new nucleotide. So here's our new strand of DNA being made. Same thing's happening down here. DNA polymerase III is adding. We see our new strands being made. Oh, there's our RNA primer, if you want to add that. But a few things to notice. Let's focus on this side since I wrote all over there. Notice how this side, the DNA polymerase is reading in this direction. Because remember, DNA polymerase can only read from the 3' to the 5' direction. So in other words, it's reading in this direction. So if it's reading in this direction and helicase is opening up, topoisomerase, they're doing this way, we're actually moving and opening as we go in this direction. So this strand here can actually be manufactured continuously, right? So it just starts, keeps going, add new bases, add new bases, add new bases, more opens up, add new bases, etc. So that is what we call one of the leading strands because it is continuously synthesized. Notice this is just a nice, even, non-fragmented line. But notice up here what's happening. Remember, DNA has to be read 3 to 5. So that means on this strand up here, DNA has to be read this way. Well, so notice DNA was read. Here's DNA polymerase I doing a little repair work. But notice that we got a new strand being made here. But when more opens up, right, because remember it's opening this direction, there's now more DNA that's going to be here that needs to be read. So because it can only move, DNA can only be read in the 3' to 5' direction, when more opens up, we have to start a new fragment and start again to make more. And that's why you see here some fragments being made. So we actually call this strand that's being made the lagging strand. It kind of lags behind. This one was leading, it was continuous, but because here we had to do it in fragments, this is called the lagging strand. Now we have a name for these fragments. They're called Okazaki fragments after the scientist who did a lot of work on this. And so this lagging strand is actually produced in fragments, whereas the leading strand is produced continuously. Now later on, what we'll talk about is that these fragments are going to have to be joined together because we don't want a bunch of fragments in our DNA. There's an enzyme called ligase, kind of like a little spot welder, that will glue those little pieces together. Okay, so a lot to take in here, but main idea from this slide is that one strand is continuously synthesized, the other is synthesized into fragments that we call Okazaki fragments. Okay, so we've added all those nucleotides, we created some strands. Now remember, we got to clean up the primer because remember there's some RNA left. So what has to happen is, here in step 4, DNA polymerase I comes in and removes sections of the RNA primer, gets rid of them, and then also replaces the RNA nucleotides with DNA nucleotides. DNA polymerase I also does some repair work if it finds some errors. Okay? So cleaning up these primers, getting rid of that RNA and replacing it with DNA is step 4. And this creates an interesting situation. So I'd like for you to think about what's happening at the end of your DNA. If at the end of your DNA, we removed a primer from one end of your DNA, that creates an interesting situation where there's a little bit of overhang, right? So if this was removed, here's some overhang. We actually call that a sticky end because other things might want to come and join to it, right? It's kind of sticky. All these nucleotides are ready to bond with other nucleotides. And this creates a really interesting situation because this overhang is not super stable, right? It's not bound to anything else. So what happens is this overhang, this sticky end, often kind of gets eroded over time and actually will shorten the DNA. Okay, so that's how we started this whole video. I said, do your chromosomes, does your DNA actually get shorter over time? What's happening here? Well, it does. But we don't really have to worry when we're young because at the end of all of our DNA are something called telomeres. Telomeres are these expendable non-coding sequences. So at the end of your chromosomes, at the end of all of your DNA are these non-coding sequences. They don't code for anything. It's just sort of like a protective cap at the end. So these are short sequences of bases repeated thousands of times, and in humans we know that code is TTAGGG. So what you see here is this TTAGGG, TTAGGG repeated over and over and over and over again. So as cells divide, as we get older and we go through replication and we go through mitosis many times, notice that the length of the telomeres gets shorter and shorter and shorter as we age. So there are many scientists who think this is one of the causes of aging because our DNA gets shorter and shorter and shorter over time. Eventually the telomere length, right, is completely, the telomere would be completely gone and we might start getting into some of the good stuff on the chromosome. Okay, so these telomeres that repeat can be removed over time, and that could cause, one of the reasons might be why we age. So step 5 then, as we wrap this up, is really we now need to go in and join these fragments. So remember, because these Okazaki fragments were created, we now need to join them back together, right? The RNA primer has been removed from there, the RNA primer has been removed from there, so now we need to join these sticky ends together. So an enzyme called DNA ligase comes and does that through a process called ligation. Oh, that makes sense. The enzyme helps these two pieces come together, and then we now have nice continuous strand of DNA. Fragments have been joined together. So this process is really quite complicated, but hopefully you now understand a bit more of how your DNA replicates itself with the help of a lot of enzymes.